Okay, well, welcome everybody, despite the weather. I appreciate those of you who could make it. Uh, February is half over. I always am glad that February is a short month. I don't know if people feel the same way. I'd like to remind you that this meeting is being live streamed on YouTube and that the recordings will be made available after the fact. We appreciate your understanding of that. Again, introduce myself, James Glazier, co-lead of the working group with Reinhard Laubenbacher. We have support from Jim Sluka, who's done web administration, a lot of scientific content as well, and then also Bruce Shapiro, who you hear from on a regular basis. If you want to contact us, we're always available to respond. Also remind you that we have a Slack. IMAG MSM is going to be pushing Slack more. We had a message from Grace saying that she wants all of IMAG MSM to be using Slack, not just us. And so again, I please encourage you to use it. And if you need access and have trouble, let us know. Also, the IMAG MSM wiki pages always need content and love, and your assistance with that would be much appreciated. I want to remind people again, next week on Monday and Tuesday, there is a workshop being held by NSF. NSF is planning some large-scale center-type RFAs on the topic of preventing the next pandemic. They're going to be interested in experiment and modeling of various kinds. The workshop next week is, I believe, mostly going to be focused at molecular and population levels. But they have Denise Kirschner speaking, doing multi-scale modeling, a variety of interesting speakers. The organizers I talked to yesterday are eager for overlap and connection with this IMAG MSM group. You can still register to hear the talks and participate, prevent-symposium.org. I think it's a good opportunity. The focus comes from rather different background. In-host modeling, I think, was not primarily their focus, but they're very interested in the connections with what we're doing. And so I would encourage people to check it out. It's going to be essentially from 10 a.m. to 5 p.m. Monday and Tuesday of next week. A reminder for upcoming meetings and mini seminars. Tomorrow morning at 11 a.m. we have our steering committee meeting for this organization. Hope I'll see many of you there tomorrow. February 25th, we have two great speakers lined up. Rita de Almeida from Brazil is going to be talking about a very interesting bioinformatic way of aggregating expression data to provide quantitative information for model validation. And Jim Fetter from Pitt is going to be talking about some of his elegant work on modeling viral replication. March 4th, we have Yi Zhang from Georgia State on the books. We have slot room for another speaker, and we're always looking for additional speakers. So please either volunteer yourself or suggest people you'd like to hear. We would really appreciate that. Now, we've been having a good time with these meetings, I think. Time always is too short. And so to try to make sure that these are maximally effective, I would say that one thing we would like is to hear, of course, to quote Yogi Berra, if people don't want to go to the ball game, nobody can stop them. So if you don't want to ask questions, you don't have to. But we have wonderfully diverse expertise in the room. And so we'd love to hear some questions from the people who have been listening so politely and silently over the past few months. And so we really would encourage people to ask questions. especially the people who have not been asking questions. And those of us like me who ask questions all the time, maybe we can hold back and let some of our more reticent colleagues have a chance. So I'd encourage us to allow that. And I also would say that because time is so limited, if we could focus on more general questions, we typically only have time for one or two questions uh, in this main session. And then what I was proposing, and I'd sort of like a show of hands perhaps as to whether you think this is a good idea, it will depend to some extent on our speakers, would be we have a very tight hour cutoff for this meeting, but we could create a set of breakout rooms for informal discussion for a half hour after the end of the formal meeting. And that would allow the speaker and people who are interested to talk freely among each other, ask more detailed questions, have getting to know you opportunities, sort of meet and greet. I would create two breakout rooms, and then after the formal meeting, I'd let people move into those breakout rooms as they request, and you'd have a half an hour. Does that sound like a good idea to people? We couldn't implement that this week because we didn't get permission in advance from our speakers, but we'd like to start that next week. And then you could have your opportunity for technical questions then. And then the other thing is it's hard to keep track of how long you're speaking. And so I'm going to give people a 5-minute warning. Just if you hear me break it and say 5 minutes during your talk, I'll do that, try to do that systematically to stay on schedule. Okay. So with no more ado, I'd like to turn over the floor to Tomas Helikar from University of Nebraska-Lincoln. We actually heard some of his mention of his work a few weeks ago, and that was a nice teaser, and I'm looking forward to hearing his talk now. Tomas.
Thank you, James. Let me just share my screen real fast. Do you see my full screen presentation mode? Okay, thank you. So thanks again for having me here today. I will talk about one of the aspects of our research program, and we're at the University of Nebraska. I'm in the Department of Biochemistry. I am a computational biologist by training, and so being housed in the Department of Biochemistry has been very interesting in a very good way, in that I've had the opportunity to work with experimental biochemists who are dealing day to day with the biological processes we're modeling here. And also it's been giving me an opportunity to slowly but surely convert experimentalists into computational modeling, or at least start dipping their toes in there. And so today I'll talk about Cell Collective, which is a computational modeling platform that we've been developing since, I guess our first paper on that was back in 2012. So for quite a bit of time. And there are a couple of reasons and sort of core principles and goals that we have set out when we decided to start building Cell Collective. And one of the most important ones is to really make computational modeling accessible to non-computational biologists. It dawned on me, or I realized that back in 2008, 9, when I was still finishing up my PhD and working with a cancer biologist, and we were building a model of a computational model of signal transduction in cancer cells to try to understand the different pathways and potential drug targets and things like that. And it became very clear that trying to communicate our modeling results with experimentalists who are not familiar with computational models, but they also may not care about the nuts and bolts of a computational model, the mathematics behind it, it dawned on me that really we need a better way to communicate models and to make models more accessible to beyond computational, the modeling community. And in doing so, it also creates the opportunity for developing these models in a more collaborative and scalable fashion. Going back to my experience again, we built this model that had hundreds of components and thousands of interactions. And I read, during my PhD, I read about 700 primary papers to put that model together. And it became very clear that if we want to get to a point where we have very comprehensive, detailed, and accurate models, there is probably not a single group that can do it alone in a reasonable amount of time. And so there is a need for collaboration if we want to make the models bigger. And we also need the models to be more transparent through annotations and providing evidence as to why a particular interaction should exist in the model or not, or why it exists in the model. And I believe that will also further give rise and facilitate reusability and reproducibility of these computational models. And so Cell Collective is a web-based platform that you can find at cellcollective.org. And over time, it's evolved. It started as a modeling technology for scientists to model different biological systems. But over the last 5 years, we've also found that, or realize that when we think about how we as scientists study biological systems through models, and then look at how we teach our biology and life sciences students about the same biological processes by having them memorize static pathways in textbooks, there is a big disconnect. And so we have expanded Cell Collective to also be a fully research or scientifically authentic educational platform. And so when you go to the landing page, you can choose to go to the research user interface or to the educational side of the interface. Today I'll be talking about the research side, and if James and Reinhard don't get tired of me, maybe they'll invite me back to talk about the education application of Cell Collective sometime in the future. So Cell Collective started as a logical modeling platform, and so probably many of you are familiar with logical or Boolean models. So I won't go into much of the detail about this, but there are some modifications that we've made to this mathematical framework that can be experienced in Cell Collective. And so in logical models, in Boolean models, every component is connected with the directed edge, and then the regulatory mechanism of that component is described with a Boolean function. So in this case, in time t+1, the component C will be active if A or B are active in time t. In Cell Collective, it is a Boolean network at any given time point, and you can see the real-time simulation, but you can also see that the output and input actually can be sort of semi-quantitative. And so what we have done is that we treat the external components as probabilities of that component being active or inactive in any time point. And so it becomes a Markov process, but you can still, the output can also still be completely binary, or it can be, as you will see in the next right here, you can look at the activity level in terms of a proportion of zeros and ones over a certain sliding window. So then you get a moving average, and that allows us to actually simulate more semi-quantitatively the impact of a perturbation on the different components of the network, or the impact of changes of dosages or activity levels of the external components. And so, as I mentioned before, making the technology more accessible has been one of the first sort of objectives that we set out to do. And so you can easily build these models basically by drawing a network, and it's building the Boolean logic all in the background. Of course, more complicated regulatory mechanisms can be defined by dragging and dropping components into as positive regulators, or developing conditions and subconditions, and the tool will construct all the Boolean logic in the background. Most recently, a couple of months ago, we actually expanded Cell Collective to also support genome-scale metabolic models using constraint-based modeling approaches, and that created new challenges. And one of them, in terms of making navigating through the models, you can imagine that genome-scale metabolic models are going to be much more complex than the current logical models that have been constructed and have been available. And so making the visualization and navigation through the web browser is very difficult. And so we've developed a new JavaScript library, WebGL library, that allows you to actually very easily and nicely, without any performance issues, navigate through very complex networks. In terms of annotations, every model in Cell Collective has its own knowledge base workspace, where you have the opportunity to annotate and also review the level of annotation for every component of the model. And so you can annotate the components at the component level. You annotate the regulatory mechanism. Every single interaction can be annotated with pieces of evidence as to why that interaction exists in the model. These little icons, by typing in a PMID or DOI, you can also go to the level of detail and indicate whether that publication is a primary source or a review article, whether it comes from human data or animal models, and so forth. And so here, actually, this model here, this network, the colors, the green color corresponds to interactions that have been annotated. If you had red or orange colors, then that would be telling you that your interactions are not very well annotated, and so gives the user a sort of higher view of the level of annotations of the models. Again, trying to make it as easy as possible for modelers to understand the extent to which they have been annotating their models. There are several different simulation tools in Cell Collective. Here is one that shows a real-time simulation, and again, trying to make modeling as accessible as possible. We provide a bunch of dials that you can increase the level of activity for the external components. You can easily select the components that you want to perturb, for example, to simulate a gain of function or loss of function feature. And then you can see the activity levels of the whole system here, or a time series graph as the simulation evolves. And you can change that sliding window that I mentioned earlier here, that gives you either, if you set the sliding window to 1, it's a purely Boolean output. If you increase it, then you get more semi-quantitative output. The fact that the external inputs are probabilities or sort of intensities of that component, of that external component, gives us the opportunity to look at input output or dose response type analyses, even with Boolean models. And so you can run hundreds of different experiments looking at the impact of external inputs on different components of the system. You can also do environmental analyses, where you can ask a question: what would happen? So the model that you've been seeing is a bacterial chemotaxis model, a very simple system. So in this case, you can ask a question, for example, what environmental condition would maximize the running motion of that system? And so it will run, again, you can base it on as many simulations. So this is simulation driven, experimentally driven. And so you run 1,000 simulations under different conditions of external inputs, and then it'll optimize, it'll find the most optimal conditions under which you will achieve that objective. And you can have an objective that's more complicated, where you want to maximize one output, but you want to minimize the other output, and so forth. Another aspect that I had mentioned before in Cell Collective is to make it enable more collaboration, and also make access to the models more easy. And so any model in Cell Collective you can export as a SBML or SBML qual for the logical models. You can also share your models directly with collaborators, much like Google Docs works. And you can also make the models available through an anonymous link. So what we've been doing is that when we submit a manuscript for publication, then we can generate this anonymous link, give it to reviewers, who then don't have to create an account in Cell Collective, and they can review the model as part of the manuscript review process without jeopardizing their anonymous status. Then to publish the models, we have a criteria that if you want to publish the model, you actually have to have every component annotated. And so I realize that maybe that will prevent some people from wanting to publish their models, but I think I would put the preference on publishing annotated models more than having more models published and less annotated. And you can also publish the experiments associated with that particular model that you're publishing. So Cell Collective, as I mentioned, because of SBML qual and SBML that also works for constraint-based models, has the ability to talk to other software tools. And so if of course Cell Collective doesn't have all the features that other tools may have, and so through SBML it's very easy to build models in Cell Collective, do some analyses in there, but then also export the models and make further explore those models using other tools. So currently Cell Collective has a little over 200 models. We're still in the process of integrating models from BioModels and BiGG Models in terms of the constraint-based models. Of these 204, there are 90 logical models that we have manually curated and rebuilt from published literature, because many logical models had been published before SBML qual even existed. And so we went back to the literature, reconstructed these models in Cell Collective, curated them such that they are equivalent to the published models, or models available in those published papers. We back annotated them because, again, as you will see, and probably even from your own experiences, you'll see that models in published models have varying degrees of annotations and supporting evidence in those papers. And so so we went back and re-annotated all the models to make it more consistent. There are a lot of different types of models. I won't talk about the COVID, but Cell Collective, Marek and Anna had already talked about the project a couple of weeks ago, I believe, about the international effort to map COVID-19. And so Cell Collective, among many other tools, has been providing the support and infrastructure for the models that the community is building. What I will talk about, 5 minutes? Okay, thank you. I will just run through a couple of other examples that are relevant to this group. And so in this particular case, this paper is back from 2012, but we have recently resumed working on host-pathogen interactions, where we use Cell Collective and our modeling approach to model the replication cycle of influenza. And so many probably know that influenza gets internalized into airway epithelial cells. The genome content gets imported into the nucleus, and then the virus hijacks the replication machinery of the host cell to produce progeny virions. And so this is relatively simple but complex or intricate process, and so we mapped it out into a logical model, and we focused on the receptor tyrosine kinase pathway that is critical for the virus to enter the cell, and looking at comparing different ways of inhibiting or intervening with the replication of the virus. We were able to we compared these different inhibitors at that time, PB1 and PA interface inhibitor, which is part of the viral replication component. That interface inhibitor was just being explored in experimentally, but you can see by being able to put the external inputs on something other than just zeros and ones, you can actually look at potential dose response curves related to inhibitors and their effect on the different components of the system. Most recently, we've resumed working on this project, and we have actually built out a much more comprehensive model of signal transduction in epithelial cells, and we have been able to identify drug targets that could target the host side of signal transduction that we're currently validating in vitro. Another project that is very recent, we have a preprint out on that, is to model the immune system as a whole at the cellular level, and so including both adaptive and innate immunity. We created this logical model that contains 10 different pathogens and 164 different components, including 35 different cytokines and nearly 30 different immune cell types. So what that now allows us to do is to look at the characteristics of different infections, and also the different characteristics of potentially different co-infections that are many times common. So to conclude, so right now our long-term goal, so Cell Collective right now supports the modeling of logical models, so signal transduction and gene regulation, but also now genome-wide metabolism using constraint-based models. We're beginning our efforts to include support for differential equation-based models, and then ultimately being able to start connecting these into multi-scale and multicellular system. And again, to kind of drive home the message, our goal of Cell Collective is to really make modeling a communication tool among the different types of scientists, ranging from basic science experimentalists to computational biologists and to translational scientists. And so I will end with that. Thank you very much.
Thank you very much. That was a lovely presentation, and I think there'll be a lot of interest. In the interest of making sure we get through the second talk, may I ask that people hold their questions until we've finished the second presentation today? Is that okay, Thomas?
Yeah, absolutely.
Okay.
Yeah.
I just want to make sure that we don't run out of time. Usually we have time, but okay. Thank you again. That was, and now our second talk is, well, you're already there, queued up. So I'll let you. I don't want to take up any of your time. I'll turn it over to you right away.
Thank you. So hi everyone, I'm Marie Ferguson. I am a project director with FICOR, which is headquartered at CUNY School of Public Health and led by Dr. Bruce Lee. and have been working with him for the last 6 years. Most of my work has previously focused on developing computational models for non-communicable diseases, largely focused on obesity prevention. But in the last year have been working with our team to do a fair amount of COVID modeling to answer a number of different questions to help with the response. So this study that I'm going to talk about today is looking at our study that explored the value of reducing the duration of the SARS-CoV-2 infectious period. So the motivation for this study really stemmed from wanting to understand if medications or vaccines that may have more subtle effects have value. We know that a lot of the public conversation in the news and media has been focused on vaccines that prevent infection or medications that completely cure the infection. And we understand that that's the ideal situation and what people are most interested in. But, there is the question that remained of, there are probably going to be vaccines and medications that emerge that have subtler effects. So what's the value of that? So one possible subtle effect that a medication or vaccine could have is, could it reduce the time that someone is shedding the virus? And if it does it, if it does, that would benefit the person, but it could potentially, reduce transmission in the broader population. So the question that we focused on was what may be the value of reducing the time that someone sheds the virus. We know that COVID-19 is a multi-scale problem that crosses the different interconnected scales. And so it's necessary to understand, how they're connected, how the effects at one scale could have downstream effects across other scales. And so that is what we aim to understand and address with the model that we developed. So this is the paper that was published in PLOS Computational Biology in January. And the paper essentially found that if you do reduce the duration of the infectious period, you can avert millions of cases and save billions of costs. So as I mentioned, we developed a multi-scale model, which represented the US population, the entire US population, so 327 million people, the mixing patterns of the population in a stochastic compartment model, and the spread of SARS-CoV-2, which we represented with an SEIR model. And then individuals that were infected in the transmission model then moved through a probability tree to basically simulate what would happen after a person gets infected. So this represented the probability of seeking ambulatory care or having a telephone consult with a provider, seeking or having to be hospitalized, and then other severe disease outcomes, which may have included ICU admission. And so as individuals travel through the probability tree and experience different clinical outcomes, they also accrued different costs in the form of direct medical costs and productivity losses. And then, of course, we represented the virus and different characteristics of the virus, including R naught, the infectious period, latent period, and relative infectiousness of asymptomatic infections. So since we were specifically looking at reducing the duration of the infectious period, we wanted to, not just represent the infectiousness as equal across the entire duration. So there were several studies coming out that were showing that viral load was rapidly increasing prior to symptom onset, and then began quickly decreasing following symptom onset for the rest of the duration of symptoms. And these figures are from 3 different studies that show, they all show on the x-axis time in comparison to symptom onset, and then some measure of viral load. And as you can see, it increases prior to symptom onset and then decreases thereafter. So how we represented this was we essentially, using data from these studies, had 43% of transmission occurring by the peak, which was at symptom onset, and then 43% of transmission occurring between the peak of infectiousness and halfway to the end of the infectious period, and then further decreasing for the remainder. So the duration of the period was on average 9.5 days. There was a distribution, it varied from 4 to 15 days. And this did include, you could transmit and be infectious the day prior to symptom onset. We also assumed that asymptomatic infections were half as infectious as symptomatic infections. So in terms of what we simulated and the scenarios that we ran, so we did assume, like I mentioned, that the average duration of the infectious period was 9.5 days. And then we ran simulations that decreased that duration by half a day, 2 days, and 3.5 days, which then corresponded to an average infectious period duration of 9 days, 7.5 days, and 6 days. And how that worked is essentially, the medication or the vaccination that we represented reduced the infectious period duration by moving individuals to the recovered bin, the recovered compartment, at a faster rate because they were drawing from infectious period duration distributions that were narrower. And then we also conducted, so we looked at the, reducing it by these different amounts, and then also treating just symptomatic individuals and then all infections, and then varied the proportion of the population that received the treatment from 25% to 75%. We also varied when the treatment started. So whether it started after 5% of the population had already been exposed to SARS-CoV-2 or after 15%, and then varied it within there. We varied the total treatment cost and the R-naught. So this is the first result slide. So what this is showing is the reduction in cases at the peak on the y-axis and the average duration of the SARS-CoV-2 infectious period on the x-axis. So here you can see, so this is at 9, this is when it was the duration was reduced by just half a day. And then this all the way here is when it was reduced by 3.5 days. So even when we only treated 25% of symptomatic cases and it only reduced the duration of the infectious period by half a day, it still reduced the cases at the peak by 1%. If we reduce it by 3.5 days, it reduced it by up to 11%. And then when we treated 75% of all infections, and reduced the period by 3.5 days, it reduced the cases at the peak by 37%. So we can start to see that even just a really small reduction in the duration of the infectious period has an effect on cases at the peak, which is important, we know, because flattening the curve is necessary to make sure that healthcare resource use capacity isn't overloaded. And I think, since what this translates to as well is since when you reduced the duration of the infectious period, and there was a reduction at the peak, you're in essence also reducing herd immunity. So the proportion of the population that has to be infected to start seeing a herd immunity threshold to start seeing a downward trend in the epidemic curve. So this slide shows the cases averted in millions. And we can see that between 1.4 and 30 million cases are inverted when the infectious period is reduced by half a day. So even if it's just reduced by half a day and only 25% of symptomatic cases are treated, there's still 1.4 million cases that are averted. And that can continue to go up even higher as more cases, people are treated, and as the reduction is increased. The other thing we see here is that there isn't as much of a benefit as from expanding treatment from just symptomatic to all infections as there is to expanding coverage. So if you can, it's more important to expand coverage from 25% to 50% to 75% than it is to expand from just symptomatic to all infections. This slide shows the results for healthcare resource use. So this is in the hospitalizations averted in millions and ICU admissions averted in millions. And so here we see that there are 3 million hospitalizations averted for when there's 3.5 day reduction in the infectious period when you treat 75% of COVID-19 cases, and then about 500,000 fewer ICU admissions, which saves greater than $757 billion in direct medical costs and productivity losses, assuming a $500 cost per treatment per person. So the other thing that I didn't mention is all these results here are assuming an R0 of 2.5 and assuming that 15% of the population has already been exposed. So this figure summarizes the results. So we can see as the reduction increases more and more and more, the cases averted increases more up to 7.4 million. This is, or at least 7.4 million when 25% are treated. And then same with costs saved as the reduction increases more and more, we see hundreds of billions of costs being saved. So the overall takeaways from this study are that reducing the time that someone sheds the virus decreases the spread. So not only does it have an impact on the individual, but it also has an impact across the population, across healthcare resource use, across their economic benefits to it as well. So I think one of the things we found is, capturing and understanding what's happening in terms of the virus, not only at the individual level, but across the population is important to really understand the downstream and the downstream effects of that. We see that even just reducing the duration of the infectious period by half a day and only treating 25% of cases reduces millions of cases and saves billions in costs. And so this can also help benchmark for researchers who are developing these medications and treatments. There may be the tendency to think that, a medication or vaccine that only reduces the duration by half a day isn't worthwhile, but we see that that's not the case, that there are still a lot of benefits that can be gained from that as well. We also see that, if there's less time to transmit the virus, there are fewer cases generated, and therefore there is a reduction in the herd immunity threshold. So for example, we saw that reducing the duration of infectiousness for 25% of cases can lower the peak of the epidemic curve by 1 to 11% and avert up to 11 million cases. This can also, the cost savings that are outlined and quantified in the study can also help, strategize how much to invest in the development of certain vaccines and medications, and also, help plan for potential different rollouts of medications and treatments that emerge that are have subtler effects. So in terms of multi-scale modeling opportunities, we are interested in continuing to develop and refine and add more detail to different scales of the model. This includes having more detailed representations and characteristics of different variants that are emerging, incorporating the effects of seasonality into the model, having more specific representations of viral shedding over time, and more detailed representations of different transmission pathways, such as aerosol transmission or transmission that occurs in the environment. And this is our team that everyone participated in developing this model. So definitely a team effort. And that concludes the presentation. So happy to answer any questions. And I think Dr. Bruce Lee is on the call as well. So if he has anything to add, can give him a moment as well.
Yeah, thanks, Marie. And thanks for presenting that. Just to add, so Marie's been heavily involved in doing a lot of non-communicable disease multi-scale modeling, but our team, and only recently over the past year or so, has ventured into COVID-19 modeling, but our team in general has been doing infectious disease modeling and epidemic modeling for the past 15 years or so. So some of the folks that she showed there have been working on these issues or problems for well over a decade and a half.
Lovely. Thank you both, for especially given how complex your subjects were, for keeping it in such a short amount of time. As usual, we wish there were more time to hear more detail. I hope both of talks suggest new collaborations. It's very interesting to see the financial and economic consequences considered in the model, and that was nice to see. So we have time now for a few minutes of questions. Again, I encourage people who haven't asked a question yet in this forum, please don't be shy and speak up. We know there's all sorts of expertise in the room, and we'd love to have your points of view. Does anyone have a specific question for either of our speakers?
Yeah, well, I'd like to make a question. That's okay. I'd like to ask Tom Helikar, what kind of experiments you do to verify your models and the results?
Yeah, so that's a good question. So it depends on the scope of the model. And the biological questions, or that drive the reason why we're building the models to begin with. In a recent paper of ours, I can share the link in the chat in a bit. We built multi-scale, we built genome-wide metabolic models of CD4 positive T cells, the different subtypes, and we were in particular looking for existing molecules or FDA-approved drugs or molecules that are in clinical trials that could be potentially repurposed for 3 different autoimmune diseases. And so when we build the models, we use the literature as a way to validate the behavior of the models, but then we also, when we have the resources, we go to the point where we test experimentally, at least in vitro, some of the predictions. And so in that particular paper, we were able to test the effect of 4 or 5 different drugs on T cell proliferation. And so it depends. Models are, computational models are much like animal models. They're just models, right? And so they won't be perfect. And so we have to, as a community, figure out the best way to communicate the accuracy, or it's not about accuracy, it's really about the usefulness of the models, right? And so is the model able to help you answer the question that you're after? And so it's a mixture of comparing your output to the literature, to independent experiments, but also to your own experiments if you can.
Okay. Thank you.
Other questions for either of our speakers?
I have a question.
Please go ahead.
To Marie. So actually I have two questions. So the first one I probably missed, but what assumption was made regarding the susceptibility to infection? Was it assumed the same for all people? This is the first question. And the second question, some data suggests that actually asymptomatic people, they shed virus longer. So did you try these different scenarios in your model? Maybe people with mild infection also will shed the virus longer.
So we assumed that there was homogeneous mixing across the population, across the entire population. So there, in that sense, it was equal probabilities of being infected. We did have age-specific probabilities of different clinical outcomes. So there were different age-specific probabilities of being, seeking ambulatory care, being hospitalized, and proceeding to more severe outcomes. And then in terms of the assumption about the asymptomatic, so we assumed that asymptomatic infections were half as infectious as symptomatic infections. So 50% less infectious. So that was a conservative estimate based on what we found in the literature. We didn't have a difference in terms of the durations between them. But yeah, so the asymptomatics were half as they're half as infectious. And so if they are just as infectious, we would have seen even greater reductions in cases averted, cost savings, et cetera.
We did account for different levels of transmission based on using distributions for R naught. So there is variability, and so we do represent variability in transmission.
Okay, thank you.
Reinhard, do you have any general business we want to cover before the end of the meeting today?
Not really. I was counting on more questions.
Okay, then by all means, let's continue the questions. I just didn't want to cut you off if you had general business.
No, I only have one remark, which takes 30 seconds. So let me just make that. As you know, most of the business that's getting done and most of the work that's getting done is being done in the subgroups. So if you have not signed up for a subgroup yet, I would very much urge you to do so. There's a large number of them available, so there should be one with a flavor that fits pretty much everybody's taste. So please sign up. That's it then, James. Thank you.
Okay, we have time probably for one last question for our two speakers. Anyone have a burning question to ask? They both kept to time, so we have time for another question. Well, if our non-usual suspects don't have questions, I will unmute our usual suspects. If our usual suspects have questions, that's fine now too.
I have a question for Tom.
Please.
So Tom, you mentioned a lot of annotations. Specifically, is the idea to make it as traceable as possible to the source of the data? Is this the usual use of annotations, or do you use them for something else? Meaning traceability?
Yes, so I look at it as, so when I say annotations, a lot of the community focuses on annotations related to IDs, like, you know, what is the gene ID of this component, and the other ontologies and things like that, and those are extremely important. But where I feel like as a community we're missing an opportunity, or I personally feel like there's not enough of, is providing the evidence as to why this interaction between two different components even exists in the model. It could be that interaction is an assumption by the model creator, or there is evidence, biochemical experiment that was done that shows that interaction does exist. But either way, wherever it's coming from, I think there needs to be more of including those in the model. So the purpose, the reason why we've created it is that to understand why we're even putting those interactions in there, and be able to trace it back as to how it fits into the system.
I guess I'd like to make a sort of a comment / question, violating my rule about not talking, which is that we have a lot of tools for designing pathways, and we have tools for executing stochastic models, probabilistic models, rule-based models. But we actually have very few tools for building what I would call either qualitative, or, to be more technically correct, conceptual models. And it's the conceptual models, that is the pathway, for example, that are portable and reproducible. And so having a tool that gives you support for annotation and model structure construction, even if there were no execution, is already very valuable. And so to me, actually, the thinking about optimal ways to do conceptual model development and presentation is something that's a really undersolved problem in computing and model building. And so I think what you've done here is very impressive. And I wonder if you have any ideas about how you might be able to extend that beyond network models to other kinds of model structures.
Beyond, well, what other model structures are you referring to?
Well, of course, interactions could always be thought of as an arrow of some kind.
Mm-hmm.
But for example, the kind of multi-scale model that we heard about in the second talk, or the spatial models that we build, these are where if you have something like cell adhesion or movement of individuals in the population.
More physics-based, yeah. I mean, it's possible, and I hope one day we'd be able to do that, and in collaboration with your tools and the tools that already do it really well. It's I don't want to reinvent the wheels in particular on the simulation and computational side of things. In some cases, because it is a web-based platform, the disadvantage that we have from the command line tools is that every feature that we want to expose to an end user has to be developed in, the user interface has to be developed for that. And so that is very expensive for us to do, because it requires, much more man hours to develop. And another aspect is web-based platform is great because it unlocks all this collaboration, but it's also very expensive when you have thousands of users trying to solve a constraint-based model or optimization problem against your server or servers. And so we've been solving that a lot by bringing the simulations into the web browser. And so all of the real-time simulations that you see in the logical model, that's all done on the user's browser in JavaScript. And so we can serve thousands of concurrent users without any strains. And so those are also additional things we have to consider when bringing in additional features and modeling approaches.
Okay, thank you very much. I'm afraid as usual we've run out of time. I hope people make new connections from this meeting, and look forward to seeing you tomorrow for the steering group, and then next week for our speakers. Thank you everybody for your patience and your participation, and very much thank the speakers for doing such a great job presenting complex material in a very brief amount of time. Thanks to everybody.
Thank you.